Okay. I'm currently a postdoctor in the Department of Biostatistics in UMC, and I'm going to give my master's report work today, and the title is Single-Cell and Spatial Transcriptomics Cluster with an Optimized Adaptive K-Nearest Neighbor Graph. And there will be 7 parts of the talk. Firstly, I'm going to give a brief introduction of the backgrounds and the details of the methods, and also the application to simulated data, real single-cell data, and real spatial transcriptomics data. And we'll also show how to use the R package we developed for this method, and finally, the summary of the work. So if you have any questions or I didn't explain clearly, you can stop me at any time. So first, we come to the background introduction of the common clustering methods for single-cell RNA sequencing data. Basically, there are 3 types of clustering methods for a single RNA sequencing data. First is k-means, and then is hierarchical clustering, and the third is community detection methods like phenograph, SERAT, and ScanPy. I think the most widely used methods are the third one, which is the community detection methods. So the process I show below is an example of community detection methods. From the initial cells, for each cell, its K nearest neighbor will be calculated. And here we show the K as 10 as an example. So only the connections for the 2 red dots are shows there. Each of them has 10 connections. And then it will have, each dot will be connected in an SN graph. And finally, there will be a cluster identified. There is only one cluster identified as the red, as the blue dots. But we can see that it potentially has a real cluster here in the red circle because these dots are somehow it's a little bit far away from the abundant cells. So it potentially has a real cluster here. The motivation of our work is pretty straightforward, that we want to know how to detect real clusters and the abundant clusters at the same time. But we also want to lose the accuracy of the abundant clusters. Another background is the introduction of some real cell type detection methods published in recent years, like Gini class 3, file, and gap class. Because we compared our results with these real cell type detection methods in our real data, So I just give some simple introduction of these 3 methods. For the Gini class 3, it combines the outcome from Gini index and final factor-based clustering to get clusters, but it always has too many clusters. So it will have a poor performance for abundant clusters. And for the second method file, it will assign a realness score for each cell. So basically, it will have real cells and abundant cells, but there is no cluster information for this method. And the gap cluster captures the huge increment of local distance to identify real clusters, but it only identifies real cluster, and it will not have the information of abundant clusters. And in the following examples, I will compare our methods with all of the 3 methods, so we'll have more clear knowledge of how these 3 methods works. And then it comes to our method. The name is aKNNO. Basically, it will have 5 steps. And for the first step, we can see there are many dots here. And in the first step, the distance of for each cell, the distance of its nearest K neighbor will be calculated. And here we just say the Kmax as an example as 10. So here, for the 2 dots, cell A and cell B, each of them has 10 connections with its 10 nearest neighbor. And in the step 2, for each of cell, its neighbors are sorted based on the distance from the small to large. And for the cell A, we can see there is a huge increasement of the distance from neighbor 5 to neighbor 6. And we also calculated a distance cutoff for each of the cell, and it's shown as the red dashed line here. So basically, in this step, we will adjust the Ka to 5 instead of 10. But for the cell B, the cutoff of distance is even larger than the biggest neighbor, which is neighbor 10. So there is no adjustment for the cell B, for the K of the cell B, and it will still equals to K max, which is 10. And I think that we will be curious how the cutoff, the distance cutoff were calculated. Actually, it's a function of the 10 neighbors and the K neighbors and parameter sigma. But honestly, we didn't get this function by ourselves. We borrowed the idea from a recent published paper in 2020, which this distance cutoff is a method for the spectral clustering, that is to build an adaptive k-nearest neighbor matrix for the spectral clustering. I'm not going to state too many of the details of this paper or of this process, but finally we can got this format for this cutoff function, which is a function. We can see that it's a function of K neighbors, distance of K neighbors, and parameter sigma. Sigma is hyperparameter to control the sensitivity to the local distance change. So it's always less than 0. And in our default function, it default to -0.5. So back to the aKNNO process and the final part of step 2. After we adjust the K for each cell and the 5 connections around cell A will be cut, and it will only left 5 connections around the cell A, and cell B is the same. And then after the SN graph construction and Loving clustering process, we finally got 2 clusters here. One is the real cluster with the red, with the red dots, and the other is abundant cluster with blue dots. And step 5 is the optimization process, and that will back to the seconds, that will repeat the process of 2 to 4. And this is optimization for the parameter sigma, because if the sigma is too small, that it means this whole function will be super sensitive to the local distance change, and it will easy to lead to the over clustering of the abundant clusters or the real clusters. So here we do a greedy search from 0 to -1 with an increment of 0.1 for sigma, and they repeat the process from 2 to 4 at each time. And we will choose the optimal sigma before there is a rapid increase of the number of the community detected. So here we can see there is a rapid increasement of the community number detection. So here we just select the optimal sigma here. And that is the whole process of aKNNO. And we can know that A means adaptive, KNNO means optimization. So this is the whole process of our method. And if you have any question, you can, okay. And then we did a simulation data set to compare our method with normal KNN. We created a simulated single cell data set from a big PBM data set, and we did 2 settings. Basically, we choose 2 cell types as abundant cell and 1 as real cells. So in setting 1, we choose the B cells and NK cells as abundant, and the real cell types are T cells. In the setting 2, the abundant cell types are NK and T, and the real is B. Actually, the setting 1 is much more difficult than setting 2 because it's really hard to distinguish the CD8 T cells from the NK cells. They are super close in the UMAP plot or in the gene expression. And we compare the for these two, for the figure A and figure B, we compare the aKNNO with KNN in different number of real cells from 2 to 20, and in different resolutions from super low, super small resolution to super high resolution. If we are familiar with steroids, if you set the resolution super high, you will get more clusters. So it will be easy for you to identify more real clusters. And if you have really low resolutions, it will has smaller clusters, and it will be hard to identify real clusters. But from the two settings to the results, we can see aKNNO is pretty stable, and it reached the perfect accuracy of real clusters, no matter what the number of real cells and no matter the resolution. But for the KNN, if the default K for the real data is equal to 20. So if the real cells is too small, they cannot identify any real clusters, even if the resolution is super high. But if it has like larger than 20, 12, or larger than 10, if it has it has high resolution, it will be easy to identify real cells. So from these two comparisons, we can say that aKNNO is much more stable and less sensitive to the resolution compared to KNN. And the figure C is an example of setting 1. We set the number of real cells as 10 for T cells. And this is a T cell, there are 2 abundant cells. And for the aKNNO and KNN with the same resolution, aKNNO can perfectly identify the 3 clusters, but KNN not. It cannot identify the real T cells. But if you want to identify the 10 T cells, it needs to go to a high resolution, and will list the overcluster of the 2 abundant cells. And this is the comparison of aKNNO with the normal KNN method in simulated single cell data. We also applied our method aKNNO to several real single cell data. Here I just show one example that from the human pancreas data set, and this 4 UMAP plot shows the clustering results from the original annotation in its original paper, and the aKNNO clustering results, and the KNN result, and also with KNN high. Here we set the aKNNO and the KNN resolution as the default resolution to 0.8, but the KNI means that the KN high means the resolution equals to 2. So it will have more clusters in this situation. And finally, we got how many? I think it's 24. Okay, sorry. But we finally got 24 clusters in aKNNO, and we compare each of the real cluster or small cluster with the original annotation, or just check the non-markers that we can confirm that what kind of cluster we have identified. For example, we can precisely identify the 3 small immune clusters like the macrophage, T cell, and mast, and also identify these small epsilon cells with 30 cells. And also we can identify a super small cluster in this part 4, which is cluster 25 with only 10 cells. It's a lymphatic endothelial cells, which is LEC, and supported by the marker of PDPN and LYVE1, the 2 non-markers, and also can show there is compared to the original annotation, it can only identify a super small standard activated stellate cluster from the abundant stellate cluster. But when we check the result of the KN and KN high, basically there has no distinguish of the three small immune clusters, and also other clusters. And besides of this non-marker, besides of this annotated cell types, we can only identify some doublets like the cluster 19. Where's cluster 19? Okay, cluster 19 here and cluster 21 here. Compared to its surrounding clusters, it shows higher number of UMI, also a higher number of genes with their surrounding clusters, and it also showed the both markers of itself and surrounding clusters, so we can identify there are true doublets. Besides, compared to KNN and KN high, we have also compared to the 3 real cluster detection method I have introduced before with our methods. The Gini class is, we only show the 10 cluster here, but actually Gini class 3 identify 85 clusters totally, and the gap class identify 4 real clusters, and the file, the yellow, not yellow, the orange here is real cells, and the blue here are abandoned cells. So totally, it identified 547 real cells in file. This is the summary of the table. Is the summary of all relatively real cell clusters we identified in aKNNO compared to all other 5 methods. Like the 3 immune clusters, we have said before that there's only Gini class 3 can identify them, but like I have said, has too many clusters and sometimes somehow it's kind of overclassed of the abundant cells. It has super large of a cluster in this part. So, and from the comparison, we can see that aKNNO perform better than all other methods in the detection of real clusters. And we have said the optimization, the step 5 optimization of sigma process. This is example to show that it's really important to do the optimization of the sigma, because our default sigma here is negative 0.5. But when we set the sigma from 0 to -1, and the y-axis is the number of community detected, and there in the when the sigma is equal to -0.8, there will be huge from negative 0.8 to -0.9. There will be huge increasement of the number of communities. So we set the optimal sigma here in the data set as -0.8. And we can compare the two results with different sigma, that we can see if we use the optimized sigma here, we can identify this doublet at cluster 21, like we have said before, and also this more the one small stellate cluster 17 here, but there will be no this kind of information in the default sigma. We also applied our method aKNNO to pan-cancer single-cell RNA sequencing data sets. There are 20 data sets from 11 cancer types, and these are the data set name. We collected the data set. We collected this data set from a database. The name is TISH. Okay, published in NAR 3 years ago, and we can see they all come from different number, and what's the purpose of aKNNO is want to identify the lymphatic endothelial cells, which is we call like LEC, and we can see these are the clusters number we have identified as LEC for each data set, and this is the cells in each LEC clusters, and the right figure is an example that how we detect and define that the cluster is a real LEC. The cluster 23 and 40 here are all endothelials, but we check the non-markers of the 2 different cell clusters that we can see in cluster 40. It has high expression of PDPN and PROX1, which are the markers for LECs. so that we said the LECs are 40, and we compare the percentage of LECs in all endothelials for each of the data sets and sorted them for each cancer type. And from this comparison, you can see that the melanoma has the highest percentage of LECs, which is consistent with the previously published paper in 2022. So that means the LEC we have identified are pretty good and are precise. So after that, we also applied our methods to 10x spatial data. We have applied to 3 spatial data, and this is one example from mouse brain. And similar as what I have shown in the single-cell RNA sequencing data, this is a cluster results for this is a raw image of the mouse brain data set, and this is a clustering result of the aKNNO, KNN, and KNN high. And when we look at the mouse brain atlas and we zoom in the 2 hippocampus structure here, and we want to emphasize that our ability to detect the precise cluster of the 2 hippocampus structure. There are 2 parts, which is ventral here, and there are dorsal hippocampus structure here, and totally we can identify 6 clusters. And for the ventral cluster, so 25 and 13 are all DG structures, but the dorsal DGs has high expression of LCT genes, and this for the CA3, which are 27 and 26, it has a high expression of IYD genes, and for the left 2 structure, which is in the dorsal, it's a subiculum structure, and in the ventral structure it's 22. Oh, sorry, this is CA1 and this is substructure. And when we compare these are all known markers that has reported in a paper that support our clustering of the 2 structures. And when we compare the same region with KNN and KNN high, we can see that they are totally messy up here, and there is no there's no any identification of these clusters. Also, like we have shown in before, that compares to other real classification methods, that we compare the 10 relatively real cluster identified by aKNNO to Gini class 3, gap class, and file. And this is a summary of the clusters, and this is the reference that we compare to the mouse brain atlas. Only KNN high has identified a small cluster here, but the Gini class 3 or gap class and file cannot identify any of the like the hippocampal structures. It's reasonable because these 3 methods are developed for single-cell RNA sequencing data, so it kind of has poor performance in the spatial transcriptomics data. But we also compared our method to other spatial and histology informed approaches, like these 4 recently published papers, which are Bayes Space, GraphST, SPAGCN, and STLearn. So we identify 31 clusters in our method, and we also use the same cluster number for the left 4 methods. So we compared the results best with the same number of clusters identified in each approach. And when we zoom in the hippocampus structure here, we can see some of them has pretty good performance, like the GraphSCT and STLearn can identify the 2 DG structure as one, and the 2 CA3 structure as one, and the 2 different CA1 structures separately, but like the Bayes space, it can only identify the 2 DJ but as one. So compared, we not only we can show better performance compared to other real detection methods, but we can also have better good performance compared to other spatial and histology informed approaches. And we also compare the running time with aKNNO that we test the running time in different number of cells and the running the efficiency in the seconds in seconds, that we show almost similar time compared to KN, but definitely we will show higher detection efficiency and accuracy of the real clusters. And this is how to use the method we have developed. Actually, it's super easy to use it. We have posted our the package in this is my supervisor's GitHub, and the name is aKNNO, and there is only one function. Only one function needs to use this method. You need to input a Serrat object. Here we name it as OB object, short name of OB object. And actually, we use the PCA matrix in this object. So it means you have to do the normalization, highly variable selection, and run PCA by yourself. And after we run the this function, and we will output is also a Serrat object, but we will store this optimized aKNN graph. The name is aKNNO. Store the graph in this name. Okay, and then if you want to get the final clusters of aKNNO, you will need to run the find clusters function in Serrat to get the final cluster, and the default resolution is the same as the final cluster, which is 0.8. And you need to emphasize that the graph name is just the graph name, the graph we have just generate. And we also uploaded several tutorials for you to from the raw data as reference if you want to try our methods and to use that using the real data. And this is the summary of this work. So we developed aKNNO, a method to identify abundant and real cell types together based on adaptive KNN graph with optimization. And we benchmark on 38 simulated and 20 single cell data and spatial transcriptomics data sets. aKNNO can identify both abundant and real cell types more accurately than those typical and specific tailored methods. And third is that aKNNO can use transcriptome alone. We don't need the histology information or location information that we will identify the structures more precisely than those spatially and informed approaches. And thanks for my two supervisors that helped me for this work, Dr. Qi Liu and Dr. Yi Sherry. And that's all. Thank you guys. If you have any questions or any suggestions, it will be super grateful. Thank you.
Thank you, Jia. Any questions, you can type into the text part, or otherwise you can just unmute yourself and ask the speaker. While people thinking of their questions, I can start with some of the questions. It's very interesting work. Thank you for presenting that. Li Jia actually have been doing related work in our group, and one of the questions I have is when you run KN and when you run your adaptive KNNO, do you have to specify the number of clusters?
No, actually, for all the data set we have applied, we use the default resolution, which is 0.8. But yeah, actually in the default resolution, you can definitely find the real clusters you want.
So when you say the default resolution, when you run the traditional k-nearest neighbor clustering, you do need to specify how many clusters you want to partition the data into. So how do you apply a resolution to the traditional k-nearest neighbor?
The same, always the same. I mean, let me show. So if we compare aKNNO with KN, that it means we use the same resolution, the default resolution 0.8. 8. But if we compare to KN high here, it means we use the resolution as 2. Yeah,
I see.
Yeah, for the simulation, it's a little bit different for the high and the default, but for the all real data in single cell and spatial, we are the same. So aKNNO and KN means the default resolution 0.8, and KNI means the K equals to 2.
Right. Any other questions?
Yeah, I have several questions. Thank you for the interesting talk. And first, I noticed that when you present your work, and most of the work is based on the UMAP plot, and to show us the difference between each of the methods. I want to know, is there any concordance matrix that you have used to benchmark across all the methods for all your data set? For example, like ARI or other matrix?
Yes, we just use ARI to compare the results, the cluster results of aKNNO and KN for abundant cells. That we want to emphasize, we didn't overclass our abundant cells, but we didn't compare ARI with other. Yeah, I think we only compare these two methods with ARI. And for the other comparison, because we want to emphasize the real cluster detection, so we just use a straightforward way, that is a table, to show that our method is best.
Okay, so another question is that I find that it seems that your method that without the spatial information is better than some of the spatial clustering method. And when you're doing those kind of analysis, have you think about that? Is there any potential reason that why spatial location doesn't help much in identify the real cell type?
That's a good question. Yeah. Reasons why they are not being so good. I think definitely I cannot ultimately I'm not sure, honestly. I'm not sure that really it is, because we only compare the 6 cluster here. It's just because before we did this clustering, we even not thought that we are the best, but we just compared the other class of other, like this spatial with histology information approaches, and we found we are even better than them. So it can be that the expression of them are super close, that the expression of them are similar, and the location of them are not. It's hard to say that. Honestly, I don't know why they are not good as expected. Yeah.
Yeah, do you think if that you can incorporate the spatial information in your method, then it will be also like have some improvement based on your current result, or maybe not?
But the I thank you, but I think if we don't use any location information here, but we are already better than them with location information. So basically we don't need to add the location information here.
Okay.
That is what we want to emphasize. We want to emphasize that we don't need information, location information. We can already be better than this approach.
So it's like,
yeah,
it's like how many like spatial data that you have tested?
Yeah, we just test 3.
Oh, only 3.
Yeah, we test 3, but we just put this one in the main manuscript and the other 2 in the supplementary. It's honestly, if the structure is more complicated, that kind of smoothly. If the structure is pretty smoothly, our method is not good, because it calculates the distance based on the distance of the spot with other spots. So it cannot smooth the layers of each spatial structure. But if sometimes the structure like this kind of irregular, this kind of some kind of strange shape, definitely our methods will be better. So that's why we only present 3, and we show the spatial result as the last of our manuscript, not the first.
Yeah, okay, thank you.
So if there are further questions, I encourage anyone who wants to stay and ask their question to continue. We have the presentation as set up as we have the meet with the speaker as the second part of this seminar. So I guess the next 20 minutes will be just informal interaction.